Hey guys. All right, we're starting to dive into the chemicals of life here. I'm going to do water and carbon super quickly. There's only a couple things on here you probably don't already know. And then maybe dive into the macromolecules. And again, there's probably only a few things on there you don't already know. So I'm going to try and rip through all that stuff really quickly. Let's see what we can do. Water, of course, is most of what's sitting in your chair right now. Most of you is water. Life happens in solution. All the stuff that happens in your body, right? Almost all of that is things dissolved in water interacting with other things dissolved in water. So like I said, life happens in solution. The chemistry of water, of course, the biggest thing about water, the most interesting thing about water is that it does this hydrogen bonding thing. It's a super tiny molecule, which is one oxygen, two hydrogens, but it has strong intermolecular forces, which are, those forces that make one molecule attracted to another molecule. So the water sticks to itself really, really well. And in fact, it has these little delta charges on there, these little small charges on each side, a negative charge and a positive charge, which makes it so it's attracted to anything that has a charge. Even if it's a very slight charge or a very strong charge, it's going to be attracted to that. It's going to be able to surround ions. It's going to be able to surround sugars, things like that. It makes it a really sticky molecule too, but it gives it a lot of interesting properties. And you've probably been through all these before, so we'll try and do them quickly. But cohesion and adhesion, this is kind of a big deal for us when we start talking about plants. And you need to know whenever you say cohesion or adhesion when you're writing a free response for AP, you should define those terms, right? Those are sciencey enough that they need to be defined. And cohesion is just water sticking to other water molecules. Adhesion is water molecules sticking to other stuff. If you think about like really tall trees, right? We've got water going, tens of meters into the air, way up there. You can't take a straw that's, say, 11 meters long and suck up water through it. You wouldn't be able to do that. Even with a complete vacuum in your mouth, if you could form that, which you can't, but even if you could, you would still only be able to lift the water about 10.3 meters. There's math that supports that. But it's pretty straightforward. But because water has cohesion and adhesion, if you make the tube really, really skinny, which is what happens in trees, right, in the xylem, which is the structure that water is transported through in trees, and we'll talk more about that later when we do transport second semester. But that takes a huge amount of the weight because the water is not just having to travel up this tube based on pressure differences. That is what gets it moving. Transpiration is the evaporation of water at those surfaces in the leaves, which is a vast surface area inside the leaves. They're evaporating a huge amount of water, and that causes water to be pulled into those vacant spots where the water evaporated away from. And so when it pulls the next water molecule isn't hanging, isn't attached to the ground. It isn't coming from the ground directly. It's coming from stuck to the tube just inside, just a little bit farther down. And right next to that, stuck to the tube because of adhesion, right, is another water molecule. And even the water molecules that can't actually adhere to the wall can stick to the water molecules that do stick to the wall. So the walls are so narrow, the tubes are so narrow in the xylem, they're basically a cell diameter, right, the size of a cell, that they are able to stick to the walls and stick to each other. And that takes a huge amount of the weight of the water. So it makes it so the plants doing this transpiration, this evaporation from the leaves, makes it so that it can transport water in this way because it wouldn't be able to pull that much weight for that entire height. This cohesion and adhesion is critical for just about any height of plant. I mean, only the smallest plants don't have to worry about that. Water, of course, is an excellent solvent. It dissolves, like I said, anything that has any kind of charge on it. So we surround our proteins with hydrophilic R groups, which makes it so that they are able to be dissolved in water, even though they're huge molecules. Salts, of course, dissolve very easily in water because they have charges. So a lot of times we divide biological substances into hydrophilic and hydrophobic. Hydrophilic means they're fine with the water. They're happy being dissolved in water. But hydrophobic are the ones that don't mix well with water. And so this allows us to have two different regions at the cellular level. In the cell membrane, we have a hydrophobic barrier. And so, there are molecules in there that are not happy to be next to water. And really, it's not the molecules, it's the water wanting to be next to itself. Water much prefers being next to something that has a charge than something that doesn't. So it's going to stick to the things that have a charge, like other water molecules, as opposed to the things that don't, that are only using London dispersion forces, if you remember your chemistry. So a molecule is hydrophilic if it has some kind of charge on it. Polar bonds are enough to do this, right? So if you have a highly electronegative atom and a less electronegative atom, those electrons are going to be shared unequally, like they are in water. And that makes it so that you have partial charges. And those partial charges are sufficient to attract water, to have them want to be by there, because that's the same type of thing that water has. This is a funny joke about polar molecules and polar bears. So, of course, a lot of biological molecules don't dissolve well in water. These are the ones that we call hydrophobic. We're talking vegetable oil, things like this, that don't mix with water. So this is a triglyceride down here. This is a fat molecule. It's what you find in things like olive oil or butter. These are actually saturated fatty acid chains, so more like what you'd find in butter or bacon grease, less like what you would find in olive oil. But we'll get to that in a minute. But they're molecules that don't mix well with water. And if you look at these things, they are largely hydrocarbons, just hydrogen and carbon in these long chains. We'll talk more about these lipids later. Ice is different from other solids because it's actually less dense as a solid. That's a little bit weird. It also makes it so that ice floats, which allows life to occur under the ice. If you think about it, if it went the other way, it would be much more difficult, because as ice precipitated down to the bottom of a pond or whatever, then suddenly the water and all the organisms living in it would be separated from the nutrients in the earth below. So that would be problematic and probably affect life in a huge way. So life can happen under the layer of ice there, and actually insulates the water from the cold air that's freezing it. So water has an extremely high specific heat. This means it holds a lot of energy, or it takes a lot of energy to change the temperature of it. And again, this is because of those hydrogen bonds. So it makes it so it's harder for the water to change its temperature. And this means a number of different things. For organisms like us that are endothermic, it helps us maintain our body temperature. Being mostly water makes it harder to change our temperature. So we actually conserve energy because it's mostly water. We don't have to spend as much energy to maintain our body temperature. It also affects climate. It means that coastal places that are much farther north can have life because it's still going to be warm. It kind of makes it so the temperatures aren't too bad. If you look at some place like Minnesota, it gets extremely cold and it gets hot in the summertime. And if you look at some place like Scotland, it doesn't get as dramatic changes in temperature, and that's because they're surrounded by water. So this, when cold air comes, then the ocean releases heat into the air and keeps it from getting too cold. And when warm air comes, the ocean takes heat from the air and keeps it from getting too warm. So it kind of like buffers the temperature. It makes it so it doesn't go to extremes very easily. Whereas if you're surrounded by land, it can go quite hot and quite cold without having a reservoir of heat that water provides. You can even see this on a very local level, because here's Palm Springs and we're looking 80 miles away. So that'd be like from here to not even Bloomington away, and you've got a 30-degree difference. And that's just because this is close to the ocean, where again all that hot air is able to dump heat into the ocean. The ocean just soaks up that heat and keeps it from being too hot, whereas there's no place to put the heat here, so it just stays and it gets nice and hot. Water also has a high heat of vaporization, so some organisms use it to prevent overheating. We do this. Dogs and other animals will pant, and they'll use evaporative cooling. They'll run a lot of blood to their tongue, for example. They'll evaporate water off of their tongue. And so when it goes from a liquid to a gas, that takes a bunch of energy to make that happen. So the energy goes with those gaseous water molecules. And so it's used for cooling. So this is kind of all that stuff on one slide. For us, the ones that are going to come up a lot, cohesion and adhesion, the fact that there's a hydrophobic and hydrophilic molecules, that's going to be important. The rest of these, maybe not so much. While we're here talking about water, let's go ahead and make sure we understand that water can ionize, right? It can split. And this is, why we have a pH of 7 in just plain water is because pH is a measure of this hydrogen ion concentration, and there is going to be some hydrogen ions. It's not very much. A pH of 7 is 1 times 10 to the negative 7th molarity of hydrogen ions. So that's 0.0000001 molarity. That's a very small amount. But that's a pH 7. We consider that neutral. That's just water by itself. Anything greater in concentration than that is going to be an acid. Anything lower in concentration than that is going to be a base. And the way you get it lower is if you mop those up with excess hydroxide. Something that can pick up hydrogens will make that even lower than 10 to the negative 7th. So we're talking about how acid or basic it is. And just to be sure you know, right, the pH scale, it is based on this hydrogen ion concentration. So when there is a concentration of 10 to the negative 2, so 0.01 molar hydrogen ion concentration, right, that's a pH of 2. So it's just the exponent there. It's actually the negative of the exponent. So that means that each level you go up, each whole number you go up or down on the pH scale, that's a factor of 10. So, that means that stomach acid here at a pH of 2 is 10 times as concentrated of an acid as the vinegar, just to give you some idea. So each one's 10 times. Now, we can't handle big changes in pH in our body. We'll talk about why later on, but proteins and things like that require us to have a pretty stable pH inside our cells and in the interstitial fluid between our cells. And that's not different from one organism to another. Everybody kind of has this same situation going on. So biological environments inside of a cell, for example, or in between cells, you're going to have buffers. And a buffer is basically a combination of chemicals that makes it so the pH doesn't change very easily. It's essentially acids with their conjugate base, or bases with their conjugate acid. It doesn't really matter. They both mean the same thing. But it makes it so that it's hard to change the pH. There's a place to put some hydrogen ions. There's a place to get some hydrogen ions in case there's a big spike. And you could chug a big bottle of vinegar and it wouldn't change the pH of your blood at all. That's largely because all of your fluids are buffers. We'll talk more about how pH can affect, life in a little bit. We'll talk about how pH affects most importantly, the enzymes, the proteins that do their jobs. I feel like I kind of rushed through the water, but I think we're okay with it. Most of that you've seen before, and AP doesn't really ask directly about it, but they do assume you know all that stuff. That's going to come up, especially, like I said, those first two things. We do need to talk about carbon, though. This is central to all of life. All of the things that happen in your body that are most interesting happen because of carbon compounds, because of long chains of carbons with stuff stuck on them. So most of you, like I said, most of what's sitting in your chair right now is water. But the part that's left is largely carbon compounds. I mean, the only thing that's left after that is some salts, really. We've got 4 types of carbon compounds. I had you name them in the last lecture. You might pause the video and make sure you can still do that. Those should be right there all the time. So we start with what's called a hydrocarbon, just chains of carbon with hydrogen on them. The simplest hydrocarbon would be methane, but as you chain more carbons along, you would have to put more hydrogens on there to make them happy carbons. Typically, hydrocarbons on their own are hydrophobic. They don't mix well with water. This is a non-polar bond for the most part. It's a very weak dipole, so we don't get a lot of charge on any part of this molecule. So they tend to be hydrophobic and not mix well with water. We take that hydrocarbon chain and we stick some functional groups on there, and this is where it gets interesting. So functional groups are things stuck to a hydrocarbon chain that give it properties of, charge, give it properties of, a different shape. There are all kinds of things that it's going to affect. Its ability to interact with other compounds come from these functional groups. So this is the list of the 6 you should be familiar with. I wouldn't say you need to memorize them or anything like that, but you should never be surprised by these things, and you should be able to know how they interact. These are the things, though, that make these hydrocarbons so different from each other and make them into things we can use to do different things. It's real easy to see how different they are biologically. Most of this molecule is the same. These 2 molecules, mostly the same. I changed this hydroxyl to a double bonded oxygen. Everything else is basically the same, except there's also another hydrogen stuck on there, and then we got a methyl group here. So some very small differences between these two. And that's the difference between a male lion and a female lion. These are sex hormones, so they have a bigger influence than some others, but it's those functional groups that make those interact with your cells, with your proteins, with all the other things going on in your body that make it so that we can have a fundamental difference like sex. So a hydroxyl group, real simple. You're probably aware of this one. This is what makes an alcohol an alcohol. You take ethane, you add a hydroxyl group to it, and you've got ethanol. Well, there's ethanol right there. So it's just an OH group. It allows it to make hydrogen bonds because now we've got an electronegative oxygen. We've got hydrogen on there. It's just like part of water, right? So we can do hydrogen bonding. This makes it really happy to be around water. These hydroxyls make molecules hydrophilic, and that's maybe the most important thing they do. A carbonyl is a double bonded oxygen. We can't get hydrogen bonding directly from this, but we can hydrogen bond to this. If we had a hydroxyl, it could hydrogen bond to this double bonded oxygen. So again, we're hydrophilic. We're going to mix well with water at this point. You don't really need to know the difference for these two, but it does come in handy. An aldehyde is when you've got on the terminal carbon, if you've got the very last carbon has a double bonded oxygen on it, that's an aldehyde. If it's one of the middle ones, somewhere in the middle of a chain, then that's a ketone, but they're still just double bonded oxygens. They're both carbonyls. A carboxyl or a carboxylic acid, it looks like a aldehyde because you've got this terminal carbon, but it also has a hydroxyl on here. So this combination means it's its own thing. It's a combination of basically an aldehyde and a hydroxyl. But this is a carboxylic acid, and we call it that because this hydrogen can come off of here pretty easily, leaving a negative charge on that oxygen, but it donates a proton, and that's what acids do. So it behaves like an acid. A lot of times we write it in a molecule like this. So here it is, as I drew it up here, and here it is after that hydrogen has come off, and now it's got a negative charge. And at body pH, a lot of these are going to be like this. You're going to be letting that hydrogen go. So a lot of them are going to have a negative charge, which means more affinity for water. It's going to have affinity for positively charged things. It's going to do other things with other molecules. An amino group, an amine group sometimes, it's just an NH2. So it looks like this. But what this often does at body pH is it'll grab another hydrogen. So you get an NH3 with a positive charge. So here's a positive charge to interact with that negative charge that we had in the carboxylic acid a little bit ago. So these kinds of things can happen. It will grab hydrogens from solution. It acts like a base, right? So this is a part of an organic molecule that will act like a base. The sulfhydryl almost never comes up. There's one amino acid that has a sulfhydryl group on it, and it forms a special kind of bond when you're doing protein folding. As the proteins fold up, it's going to actually form a covalent bond between these sulfurs. But it's not something you need to worry about too much. It doesn't really come up often. It's really just a way to stabilize proteins, and we'll get to that here in a bit. Phosphates come up a lot when we're talking about nucleic acids because phosphates are part of the backbone of DNA and RNA. We already talked about it already in the last lecture when we were talking about ATP. It was 3 of these guys, right? TP, triphosphate. But they have negative charges. They've got lots of oxygens, lots of polar bonds, happy to interact with water. But this phosphate is something we stick on a lot of molecules. We take off a lot of molecules. So a lot of times we have proteins that are inactive, and then you stick a phosphate on there, and now they're active and they do their job. And when you need them to stop, you just take the phosphate back off and they go back into their inactive form. So that gives us like a reservoir of enzymes or proteins that can do a job that we can turn on when we want and turn off when we want very quickly. So we do that a lot with phosphates. And of course, like I said, they're in those high energy molecules like ATP. If you have questions, you should definitely ask. You should ask your neighbor. You should ask me. You can shoot me an email. You can wait till Mondays and when we meet, and you can ask me then. But you do need to ask questions. Now we're going to talk about carbs and lipids. We'll leave nucleic acids for second semester when we do genetics. We'll do proteins, and that'll be an entire presentation on its own because proteins are kind of a big deal. So carbohydrates and lipids, and then we'll be done with this one. So looking at carbohydrates, carbohydrate. When you see ate in a chemical, that usually means an oxygen compound, chlorate, carbonate, nitrate, right? Those are all oxygens with the nitrogen or the chloride or the carbon. Hydra, of course, is hydrogen, and carbo, of course, is carbon. So carbon, hydrogen, and oxygen. So usually they're in a 1 to 2 to 1 ratio. So the empirical formula is going to be something close to that. Sometimes there's an oxygen fewer or maybe a couple of hydrogens off, but it's going to be pretty close to that. And our primary use for this is energy. This is our usable energy. This is the bulk of our diet is carbohydrates, especially here in the United States. But a tiny fraction of our actual body mass is carbohydrates. And that's because we burn it for fuel very quickly. So it's mainly for energy, both short-term storage and for transporting energy around. But this is the reason we consume it. This is the thing that plants are going to be making. They're going to start by making carbohydrates when they do photosynthesis, and then they will use those carbohydrates to actually a short carbohydrate to make all the rest of the things they want to make too, including amino acids and nucleic acids and things like that we will eventually want. Lipids too, of course. We also use these, both plants and animals use these for structure. I should say plants, animals, bacteria, everything else. A lot of extracellular stuff. So cell walls in plants, or we have a layer called a glycocalyx, a bunch of chains of sugars that are outside of most of our cells that are mostly for cell identification, but also provide some structure. Bacteria will have cell walls that are made of various carbohydrates. The exoskeleton of all our arthropods, that's all chitin. The cell walls of funguses, those are chitin. Those are all carbohydrates. So lots of structural stuff too, but primarily energy. Usually with sugars, we end up with a suffix that is OSE, and we sort them out by numbers of carbons a lot of times. So, there's glucose, ribose, and glyceraldehyde. It would be glycerol if it was a hydroxyl group here, but pretty much the same thing. So this would be a triose with 3 carbons. This is a hexose, and this is a pentose. So they're basically given a prefix based on how many carbons there are, and then end in OSE. When we have carbons in a ring structure like this, we number them and so that we can identify which carbon we're talking about. And whenever it's in a ring like this and you've got a bit dangling off like that, we number them starting on the ring. So our number 1 prime carbon is this one here. And then we have the 2 prime, 3 prime, 4 prime, 5 prime, 6 prime. It doesn't really matter for a sugar like this, a hexose like this. It'll matter when we look at deoxyribose or ribose, because those are kind of a big deal with the structure. We need to look a lot more closely at the sugar when we're looking at nucleotides and DNA and RNA. So that'll come later, but just as a heads up. Monosaccharides, simple sugars, glucose, fructose, galactose, right? These are our ones that we can absorb easily through our intestinal wall. They're just a single monomer, basically. You could stick 2 of these together and have a disaccharide. Maltose is a good example. That's just 2 glucose. Lactose is galactose and glucose. Sucrose, which is table sugar, is a fructose and a glucose. So those are disaccharides. Like I said, sucrose is how plants usually move their energy around. They use sucrose. We usually use a monosaccharide, glucose, to transport energy around inside our bodies. Lactose, if you're lactose intolerant, the thing you can't do is break apart the galactose and the glucose. You can absorb either one of those, but together you can't absorb them. And so if you can't break that, then that sugar goes on past your small intestine and into your large intestine where there's lots of bacteria, and you're feeding those bacteria a lot more than what they're used to. So you get gastrointestinal distress. Basically, the bacteria start reproducing like crazy because they're getting huge amounts of food that they don't normally get. And that causes a lot of problems. Polysaccharides, we're talking starches, glycogen, cellulose is one of these, chitin is one of these. So they're chains of simple sugars, long chains of them, of repeating units. This is a polymer. So these are polymers of simple sugars. Starch is a polymer of glucose. Cellulose is a slightly different polymer of glucose. Glycogen is a branchy polymer of glucose. These are all chains of individual glucose molecules. When we go to polymerize these, of course, this is a job for an enzyme. We can take monosaccharides and put them together to make disaccharides. And of course, we use dehydration synthesis. We're going to take a water out of here. There's our water coming out. Like I said, some polysaccharides we use for structure. Cellulose in plants, the cell walls of plants, this clear part here, that's very sturdy, very strong stuff. It's what, it's what wood is made out of. It's strong stuff. And we'll talk a little bit more about that later. Chitin is the exoskeleton of arthropods. It's the cell walls of funguses. The other reason we polymerize glucose is to store it. And so plants do this forming a molecule called starch, which is just long chains, maybe a few branches, but not very branchy, basically long chains of glucose. When we make our animal starch, which we store a lot in our liver, we do have it in our muscles, so it's readily available too, but lots of it banked up in our liver, the bulk of it. And this is the sugar that'll be released if your blood sugar drops down, right? So if your blood sugar goes up, you store it in the liver. If your blood sugar goes down, you release it from the liver. When it's in the liver, it's stored as glycogen, which is chains of glucose, but they're really, really branchy. So let me show you what I mean. So this is fairly typical of a plant, right? Or maybe, like I said, a few branches, but not very branchy, whereas animal starch is very branchy. Glycogen, which is what we call animal starch, is very, very branchy. And so the reason for that is because sometimes we're in a hurry, and a tree is never in a hurry, right? A plant never needs a huge amount of energy right now. Even if, they're in their most perilous moment, they can't hurry away. They don't have the capacity. They don't have the ability to move at all. The enzymes that are going to break these starches down just take the terminal glucose. So if you got long linear branches, you can only take one off the end at a time, and then you can take another and another and another. But if you got all these branches, look, you can grab every terminal glucose on this thing, and there are tons of them, and you can grab them all right now. So the amount of sugar you can make available in your blood quickly is huge. You can get a huge amount of sugar. And this is what happens when adrenaline is released, for example. You'll dump a bunch of sugar into the blood so that you're ready to do, fight the tiger or whatever it is you've got to do. A plant's never in that situation. So, having a huge rush of sugar is not going to help it do anything anyway. So, like I said, one little tweak in the structure of this, we can make alpha glucose or beta glucose. And as we chain them together, in starch, we use this alpha glucose. All kinds of organisms have an enzyme that can break down chains of alpha glucose, but very few organisms have enzymes that can break down chains of beta glucose. Starch is very easy to digest by just about everything on Earth, and cellulose is very difficult to digest, and the enzymes to do that are very rare. So we have enzymes to break down starch. We do not have enzymes to break down cellulose. Cellulose is an extremely abundant compound. It is everywhere. It's in all plants. It's the structure of all plants. It's the bulk of all plants. Huge amounts of carbon stored in this stuff, which is, what gets released as carbon dioxide when we burn plants, when we burn trees, when we burn paper, when we burn any of these, wood or plant-derived materials. There are some organisms that simply eat the tender parts of plants that are not heavy in cellulose. There are other organisms like cows and rabbits and things like that that can eat large amounts of cellulose and get energy from it, but they have symbiosis going on that helps them. They're not actually able to digest it. They have somebody inside of them that can. Same with termites. For us, though, this is fiber. This is undigestible. It goes right through us. We call it dietary fiber, but it's cellulose. It's wood. It's just not quite as hard as wood in the plants that we eat. But you may have noticed, like corn, when you eat corn, sometimes you see it again. The reason you see it again, what you see are kernels that you never broke the outer boundary of. If you didn't bite into it, then your enzymes can't get into the inside part because that outer layer of cellulose is indigestible to you. So that'll go all the way through without ever getting to the inside of it. So it'll still be yellow and everything. You have to break through that with your teeth to be able to get into the inside of that and not have that show up again. If you look at cows, they have a robust digestive system, much more complicated than ours. And they have a huge fermentation tank in there, and they pass food into it and back out and chew it again and pass it back in again, over and over again to get huge amounts of surface area for these bacteria to digest that cellulose. And then they can digest these big mats of bacteria that are getting energy from the cellulose. So it's like a little food chain inside of them. Termites are the same way. Termites actually have several organisms. So they put wood in there that they are unable to digest. And they have protists that live in their gut, but the protists can't digest wood either. They have bacteria that live in the gut. The bacteria can digest the wood. the protists eat the bacteria, and the termites digest the protists. And so there's a whole food chain going on. So that means that a termite's first meal has to be the poop of another termite so that it can be inoculated with both the bacteria and those protists. Kind of gross. Another one of those things that makes you appreciate, things we don't have. Rabbits, speaking of things that are gross, rabbits have two kinds of poop. They poop once, and it's kind of a pasty, greenish poop, and then they eat that, and then they poop again, and it's a more pellety, dry, most of the energy, well, maybe not most, but a lot of the energy is extracted from it. All these guys, herbivores, have poo that is still high in calories, still has lots of energy. So that's why we have things like dung beetles that are able to make a living off of herbivore poop, because there's still a lot of energy in there, because it's very difficult to get all the energy from this. So that's carbohydrates. I'm going to move on to lipids now, which are, generally speaking, lipids are hydrophobic biological molecules. And so they're not all exactly alike, or even there are some that are pretty different, but they are all hydrophobic. That's the characteristic they have in common. So mostly carbons and hydrogens, but there is oftentimes a little bit of oxygen, occasionally a nitrogen, things like that. But for example, this is a kind of a typical fat molecule, and you can see there's lots and lots of just hydrocarbons out here. And then there's oxygens over on this glycerol part of this backbone part of this. They come in three main groups, and they are triglycerides, phospholipids, and steroids. So there are a couple of other groups that we didn't include here. Waxes would be in here as well. And there's some other compounds, but those are, we barely ever talk about them. So these are not going to form polymers. They're not going to be repeating units. They may be brought together of multiple units. For example, in the gray box here, I have a glycerol, and then I have 3 fatty acid chains here to make this triglyceride. So those are 4 different components that come together to make one molecule, but they're not strung together one right after another, like the glucose was in starch as a polymer. They're just, big molecules with smaller subunits. Triglycerides, fats, oils, things like that. What makes olive oil different from butter, things like that, is what these chains are like. But they are typically all triglycerides. So we got this glycerol here, which is a triose. It's a 3-carbon sugar, right? And then each of those carbons has an oxygen bound to it. And that's what glycerol is. It's 3 hydroxides is what it would have. We're going to do dehydration synthesis to attach a fatty acid chain to each of these. So there would be a hydroxyl right here off of this fatty acid chain, because fatty acid, this is what makes it fatty, the hydrocarbons. And the acid, it's a carboxyl group. So there's your double bonded oxygen. Remember, it had a aldehyde, and then it also had a hydroxyl. We took the H off the hydroxyl, and then this had a hydroxyl on the carbon. So we took 2 H's and 1 of the oxygens to make water, dehydration synthesis. You can see that right there. So when one of these is made, is water formed or broken? That you should remember. And how many water molecules? So pause the video and see if you can answer that. So you can see right here, we've got 3 places where we're going to do dehydration synthesis when we make it. So dehydration synthesis means water comes out. So we're going to produce 3 water molecules when we make a triglyceride. And when we break this thing down into fatty acids and glycerol, which happens in our small intestine, when we do that, we're going to have to put in 3 water molecules. So these are really big molecules, and they tend to be almost entirely nonpolar. There are some polar bonds in here. Those oxygens are going to have polar bonds. And so it's going to be a little hydrophilic on this end. But the bulk of the molecule is hydrophobic, nonpolar bonds. And it's going to be things like long-term energy storage. That's our main function here, is long-term energy storage. This is not something you can get too quickly. You can't even, if you go run a marathon, you're not going to be burning fat. What you have to do is establish a pattern of caloric deficiency, meaning you're not taking in as many calories as what you're using day after day after day before your body gets the signal, right? It's a kind of a hormonal thing that says, okay, we need to start breaking down some of this fat. So you're not going to burn it exercising. That's not the way it works. You have to set up this deficiency that goes over time to get the body to make this transition away from depositing fat to breaking down the fat. Lots of organisms use this as insulation, right? In the water, hair doesn't work very well because hair gives you a layer of trapped air that works as insulation so that cold air outside can't get, can't take the heat from your body because there's this barrier. But in the water goes right through that barrier, so it doesn't keep you cool. So an adaptation for staying warm, for warm-blooded organisms that live in water, they're going to have a layer of fat. So, whales and dolphins and seals and stuff like that, walruses and whatnot, they've all got a lot of fat. It is kind of interesting to note, I think I've mentioned before, the aquatic ape hypothesis. One of the things that we have that's very different from our closest relatives, the chimpanzees, is this layer of fat that's all over our bodies. And it is interesting that it is a characteristic that we share with aquatic mammals. This is a good question the penguin asks. This is a huge source of calories. This carbon has more energy than a carbon in a sugar, for example. So this is super dense, like ounce for ounce, twice as many calories as what sugar would have, or starch. So very high in energy. So liking these things is an evolutionary, right? It's a trait that makes it so that you are seeking these things out so that you are getting enough energy. Fats come in a couple of different types, saturated and unsaturated, and really that's about these tails. Saturated fats have long, straight tails. They don't bend. They lay together, stack very nicely side by side like this. And that's because every one of these bonds in these carbons are single bonds, and single bonds are able to swivel. And so being able to swivel around makes it so that water can get out from between there and they can lay really close together. This is generally what you find in animal fats. They're solid at room temperature. They'll be more fluid at body temperature. None of these bonds are double bonds. The double bonds between carbons, they form a pi bond. The pi bonds don't swivel, so they end up locked in a particular position. And if you've got more than one of these things, you're going to end up in a position where you can't lay out real flat like this. And those are unsaturated fats. They have what we call kinky tails. And that's because there's double bonded carbons in here. If you think about this, right, you've got a carbon double bonded to another carbon. Let's just imagine there's more carbons out this way, right? You're in the middle of the chain. This guy has a hydrogen on it. This guy has a hydrogen on it. This can't swivel. If we wanted to make it so it was a single bond, what we would have to do, because each of these carbons now have four things attached to them, which is what makes carbon happy, right? It's got two bonds here and two single bonds. If we bubble hydrogen gas through here, what happens is this bond will break and the hydrogen will react and stick here, and the hydrogen will react and stick here, and now we got single bonds. So we can take these kinky-tailed unsaturated fats, bubble hydrogen gas through them, and we can make them saturated. So these, because they don't lay down real nice together, they don't form a solid at room temperature. They are always in motion, so it's a liquid. But if you bubble that hydrogen gas through there, then suddenly these bonds can swivel and they'll line up side by side to exclude water, and it makes it so that they can stack together much more tightly, and you get a solid at room temperature. So that's what hydrogenated vegetable oil is. You do have to be careful when you do that, because sometimes you end up with what are called trans fats. We don't really need to get into that, but those can be solid not just at room temperature, but at body temperature. And that's a problem because in your blood vessels, you end up with this solid fat, this solid lipid building up. The oils in fish, the oils from plants, these are all going to be unsaturated. And they are considered healthier because, again, they don't stack up and form plaques as easily in your body. And these are the ones that make up cell membranes. So we have two kind of parts of this molecule. It's got hydrophobic tails, and it's just based on that same glycerol molecule, only instead of having 3 fatty acid chains, we have just 2. And then there'll be a phosphate and then some other stuff. And a lot of times that stuff has charges on it. But it makes it so this top part, this is all hydrophilic. The bottom part is hydrophobic. So we have 2 different sides of this molecule. Sometimes we say it's got a hydrophilic head and then 2 hydrophobic tails. And a lot of times we'll just draw it like that simple little diagram like that. That represents a phospholipid. And so as soon as you have these things, you throw them into water and they will spontaneously line up because water's trying to get next to water instead of those guys. So they'll line up side by side, a whole bunch of them with their tails together to form this membrane. And now the bottoms of that are exposed to water, and they don't like that either because they're hydrophobic. And so you take another membrane like that and put it on the bottom, and now you've got this bilayer, this phospholipid bilayer. So you can see the tails are all aligned in here. This whole area inside of here is all hydrophobic. Water is being excluded from there. These parts of the molecules are totally happy to be next to the water outside the cell or inside the cell. So they will spontaneously form these membranes just by putting these molecules in water. So when we assemble these, we are assembling membranes because they automatically pop in and make more membrane. And the cool thing about that is that now we've got inside, like this is me and everything else is outside. So it gives us a boundary to the organism if you're a single-celled organism, or a boundary to a functional unit in a multicellular organism. Oh, soap, yeah. Soap's like this too. Most of your detergents have a hydrophilic side and a hydrophobic side. So if you get, grease on your clothes, you add laundry detergent to it when you wash it, and the hydrophobic side will go to, if the grease is like, bacon grease or whatever, it'll be a triglyceride. The hydrophobic side will go to that triglyceride, and there'll be a bunch of them that all surround a triglyceride with their hydrophobic end. And now the outer layer of this thing you just made is hydrophilic. So you can wash that away in the wash. And so it'll surround one by one these triglycerides, or clump by clump, and take them away. Bile works the same way. We have what are called bile salts in our digestive tract, works to separate fat globules. Because otherwise, as soon as you ate a bunch of French fries, as your body churns, as you're in your stomach and small intestine, as you churn that all up and you make it into all a liquid, because you need everything in solution, the fats are all going to clump together in one big clump, and you're not going to be able to digest them all. So what this bile salts do is they separate it into smaller droplets so that the enzymes that break down the fats can get to every single one of them, and you don't pass a bunch of energy down to your large intestine, like what we were talking about before with the lactose. The last group is steroids. These are all cholesterol derived. We start with cholesterol, and then you change functional groups on here, add functional groups of various kinds, and you get a bunch of different steroid hormones, which are hydrophobic molecules that signal cells to do things. So we've got these 4 carbon rings. Like I said, a variety of different functional groups. Cholesterol in itself is something that animal cells use. We don't have cell walls. And so we need something to make our cell membranes a little more rigid, but also we need to keep them fluid in extreme conditions. So at high temperatures, they need to be strong enough to not fall apart. And at low temperatures, they need to stay fluid because animals encounter these situations. And so we use these in our cell membranes to kind of stabilize them. It makes them sturdier at high temperatures, more fluid at low temperatures. It just makes it more useful in a broader range. And it also makes it a little bit sturdier just in general because we don't have a cell wall. It makes it so our membranes are not so easily disrupted. A plant cell or a bacterial cell or even a fungus cell, they have the cell membrane, but right up against it is a cell wall that gives it some structure and gives it some support. We don't really have that in animal cells because of this ability to move around. And so to keep the cells kind of intact, we add a little cholesterol and it stabilizes those membranes. It can, like any other lipid, it can build up in blood vessels and cause problems. So it is an issue, especially when you eat a more American-style diet with lots of lipids. So like I said, we just make a few tweaks and we can get a variety of different hormones. These tend to be sex hormones, right? The things that, cause puberty, make you grow a beard, things like that. These are not high-speed, real fast-acting hormones. It's not like adrenaline. It's not going to be a situation where there's a tiger and you have to grow a beard really fast. That's not what happens, right? If there's a tiger, you release adrenaline that helps you, release body sugar, blood sugar, gets your muscles ready to fight, gets your, heightens your awareness, does all this stuff that adrenaline does. Sex hormones are much slower. They work over years of time. But they make differences of all kinds of different organisms, right? This is why a male peacock looks different from a peahen, which is what the female is called. So it's just those functional groups stuck on cholesterol. So that's lipids. You need to be pretty familiar with them. Hopefully you've seen carbohydrates and lipids before and you remember it a little bit. Monosaccharide seems to be something kids remember, polysaccharides, things like that. But we're going to be talking a lot about these molecules, so you got to be familiar with how they work and what they're used for. Hang in there, guys. You're doing great.